Hello friends, today we will discuss about cell. Details about cell. Let's study about the models of membrane structure, and here we can discuss about an experimental perspective. Okay. So, until electron microscopy was applied to the study of cell structure in the early 1950s, no one had ever seen a membrane. Yet, the indirect evidence led biologists to postulate the existence of membranes long before they could actually be seen. In fact, researchers have been trying to understand the molecular organization of membranes for more than a century. Because cells contain many different kinds of membranes, finding the structural features that are common to all membranes has been a special challenge. The intense research effort paid off, however, because it led eventually to the fluid mosaic model of membrane structure. This model, which is now thought to be descriptive of all biological membranes, envisions a membrane as a fluid sea of phospholipids and other lipids with proteins floating in and on it. Before looking at the at this model, we will consider the series of historical developments that lead to our present understanding of membrane structure and function. As we do so, you may also gain some insight into how such developments come about, as well as a greater respect for the diversity of approaches and techniques that are often important in advancing our understanding of biological phenomena. Now let's know about the Overton and Langmore. Lipids are important components of membranes. A good starting point of our historical overview is the pioneering work of Charles Overton in the 1890s. Overton was aware that cells seemed to be enveloped by some sort of selectively permeable layer that allowed different substances to enter and leave cells at significantly different rates. Working with cells of plant root hairs, he observed that the lipid soluble substances penetrated readily into cells, whereas water soluble substances did not. In fact, he found a good correlation between the lipophilic, that means lipid means loving nature of a substance that the case that there is with which it could enter cells. From his studies, Overton concluded that lipids were present on the cell surface as some sort of coat. He even suggested that cell coats were probably mixtures of cholesterol and lecithin, an insight that proved to be remarkably foresighted in light of what we now know about the prominence of sterols and phospholipids as membrane components. A second important advance came about a decade later through the work of Irving Langmuir, who studied the behavior of purified phospholipids by dissolving them in benzene and layering samples of the benzene lipid solution onto a water surface. As the benzene evaporated, The molecules were left as a lipid monolayer on the water surface. Langmuir knew that phospholipids are amphipathic molecules with hydrophilic heads, hydrophobic means tails, non-polar tails. He reasoned that phospholipids orient themselves on water such that their hydrophilic heads face the water and their hydrophobic tails protrude away from the water. Langmuir's lipid monolayer became the basis for further thought about membrane structure in the early years of the 20th century. Now let's study about the Gotter and Grendel. The basis of membrane structure is a lipid bilayer. The next major advance came in 1925 when two Dutch physiologists named G. Gotter and Grendel read Langmuir papers and thought that this approach might help answer a question regarding the surface of the red blood cells or erythrocytes with which they worked. Overton's earlier research had shown the presence of a lipid coat on the cell surface, but how many lipid layers were in the coat? To answer this question, Gotter and Grendel extracted the lipids from a known number of erythrocytes and used Langmuir method to spread the lipids out as a monolayer on the water surface, finding that the area of lipid film on the water was about twice the estimated total surface area of the erythrocyte. They concluded that the erythrocyte plasma membrane consisted of not one but two layers of lipids. Assuming a bilayer structure, Grendel and Gotter reasoned that it would be thermodynamically favorable for the nonpolar hydrocarbon chains of each layer to face inward, away from the aqueous milieu on either side of the membrane. The polar hydrophilic group of each layer would then face outward toward the aqueous environment on either side of the membrane. Their experiment and their conclusion were momentous because this work represented the first attempt to understand membranes at the molecular level. Moreover, the lipid bilayer that they envisioned became the basic underlying assumption for each successive refinement in our understanding of membrane structure. Now let's study about the Debson and Daniel Lee. Membrane also contain proteins. What they say? They say membranes also contain proteins. Many perplexing contrast between artificial bilayers and biological membranes soon made it clear that the lipid bilayer, though an essential feature of membrane structure, could not explain all the properties of the membranes, particularly those related to surface tension, solute permeability, and electrical resistance. For example, the surface tension of a lipid film was shown to be significantly higher than that of cellular membrane but could be lowered by adding protein to the lipid film. Moreover, sugars, ions, and other hydrophilic solutes moved into and out of cells much more readily than could be explained by the permeability of pure lipid bilayers to water soluble substances. For example, the leakage rate of potassium ions across artificial bilayers is measured in days, whereas the same amount of leakage across erythrocyte membranes occur in an hour or so on. To explain such differences, Davison and James Danieli invoked the presence of proteins in membranes, proposing in 1935 that biological membranes consist of lipid bilayer coated on both sides with thin layers or lamella of protein. Thus, the original Davison Daniel model was in essence of protein lipid protein sandwich. It is sandwich. This model was the first detailed representation of membrane organization and dominated the thinking of cell biologists for the next several decades. The original model was subsequently modified to accommodate additional findings. Particularly notable was the suggestion made in 1954 that hydrophilic proteins might penetrate into the membrane in places to provide polar pores through an otherwise hydrophobic bilayer. These proteins could then account for permeability and resistivity properties of membranes that were not easily explained in terms of lipid bilayer alone. Specifically, the lipid interior accounted for the hydrophobic properties of membranes and protein components explained their hydrophilic properties. The real significance of the Davison-Daniel model, however, was its recognition of the importance of proteins in membrane structure. This feature, more than any other, made the Davison-Daniel sandwich the basis for so much subsequent research on membrane structure. Now let's study about the Robertson. All membranes share a common underlying structure. What he says, Robertson says, all membranes share a common underlying structure. All of the membrane model we discussed till now were developed before anyone had ever seen a biological membrane, and each was intended specifically as a model of plasma membrane. With the advent of electron microscopy in the 1950s, cell biologists could verify directly the presence of a plasma membrane around each cell. In addition, they observed that most subcellular organelles of eukaryotic cells were also bounded by membranes. Furthermore, when membranes were stained with osmium, a heavy metal, and then examined closely at high magnification, they were found to have extensive regions of railroad track structure that appeared as two dark lines separated by a lightly stained central zone with an overall thickness of 6 to 8 nanometer. This pattern is seen for the plasma membranes of two adjacent cells that are separated from each other by a thin intercellular space. In fact, the same trilaminar or three-layer staining pattern was characterized of many different kinds of membranes, led J. David Robertson to suggest that all cellular membranes share a common underlying structure, which is called the unit membrane. When first proposed, the unit membrane structure appeared to agree remarkably well with the Davison-Daniel model. The two dark lines were equated with the phospholipid head groups and the protein layers on either side of the membrane, which appeared electron dense because of their affinity for heavy metal stains. Now let's study about the Singer and Nicholson's fluid mosaic model. What this Singer and Nicholson's a membrane consists of a mosaic of proteins in a fluid lipid layer. The fluid mosaic model proposed in 1972 by S. Jonathan Singer and Garton Nicholson has two key features, both implied by its name. Simply put, the model envisions a membrane of as a mosaic of proteins discontinuously embedded in or at least attached to a fluid lipid bilayer. In other words, the Singer-Nicholson model retained the basic lipid bilayer structure of earlier models but reviewed membrane proteins in an entirely different way, not as flat sheet on the membrane surface but a discrete global entities that associate with the membrane on the basis of their affinity for the hydrophobic interior of a lipid bilayer. This way of thinking about membrane protein was revolutionary when first proposed by Singer and Nicholson, but it turned out to fit the data quite well. Based on differences in the nature of their linkage to bilayer, three broad classes of membrane proteins are recognized. Integral membrane proteins are embedded within the lipid bilayer where they are held in place by the affinity of hydrophobic segment on the protein for the hydrophobic interior of the lipid bilayer. Peripheral proteins, on the other hand, are much more hydrophilic and are therefore located on the surface of the membrane where they are linked non-covalently on the polar head group of phospholipids and to the hydrophilic parts of other membrane proteins. Lipid anchored proteins, though not a part of the original fluid mosaic model, are now recognized as a third class of membrane lipids. These are essentially hydrophilic proteins and therefore reside on membrane surfaces, but they are covalently bound to lipid molecules that are embedded within the bilayer. The fluid nature of the membrane is the second critical feature of the Singer-Nicholson model. Rather than being rigidly locked in place, most of the lipid components of a membrane are in contrast constant motion, capable of lateral mobility. Many membrane proteins are also able to move laterally within the membrane, although some proteins are anchored to structural elements on the one side of membrane or the other and are therefore restricted in their mobility. The major strength of the fluid mosaic model is that it provides ready explanation for most of the criticism of the Davson-Daniel model. For example, the concept of proteins partially embedded within the lipid bilayer accords with well the amphipathic nature and globular structure of most membrane proteins and eliminates the need to accommodate membrane proteins in thin surface layers of invariant thickness. Finally, finished any questions regarding this, you can ask me in the comment box and subscribe my channel to know more updates. Thank you.